Okay, we're live,
We
boys
are.
and hyenas. I have a picture of a hyena for today's show because I thought it was appropriate.
Yep.
Because Danny was even talking about the hyenas being after him, and I think he was referring to us.
Where's Leo when you need him?
I don't know where Leo is.
Hopefully we don't have John, our resident boomer, coming in and harping on Leo for whatever.
I emailed him. Oh, there's four now. I'm not sure if anybody's watching in chat or not. Let's see here. Save chat. Where am I? What's happening there? Anyway, so Standing for Truth did a big comeback against Dr. Dan tonight. Actually, I can't use theater mode. Yeah, can't do it. I got to do something else. Where's default view? Okay. I'm going to go back over here. Okay. So I only listened to a little bit of this, but I kind of wish Walker was here because I'd like him to address what Standing is saying here. Kind of interesting. Hey, Leo, come on in.
You know, it doesn't seem like Standing makes too many new points, really, from what I've observed of him.
No, which is interesting because he's claiming to demolish.
But is it just me, or does he kind of like shift in terms of which claims he wants to resuscitate and claim have been refuted or haven't been refuted? That was a question.
But was it a question?
Yeah, I was asking, is it just me, or does he kind of like shuffle between a set amount of talking points and kind of arbitrarily choose or determine like when one's been debunked for the time being and when to resuscitate? Kind of an old argument.
Yeah. Listen to this a little bit here.
Okay. The fact is, we actually know Eve. Okay. There is no debate here. We know her sequence.
Okay. So he's been saying that for a few shows now, but he claims to know Eve's mitochondrial sequence. And then he goes on to explain how they get it. And
is he deriving mitochondrial Eve in the same way that we are?
Well, they're
like using mitochondrial clocks.
Yeah, he claims to know Eve in a different way, but let me play it and see if it makes any sense to you at all. Maybe it does, but I didn't quite understand.
It's been published, and the critics don't want to read the papers that we cite in support of our position. Okay,
we don't read the papers. His critics write those papers. The people that study evolution are the ones writing the papers. And this is...
well, I'm going to explain then in a nutshell, how we know her sequence. Mitochondrial DNA, being the uniparentally inherited DNA, is only about 16,000 letters long. So all we have to do is take all the mitochondrial DNA in the world. We can line them up. And when we line them up, what we realize... we understand that 85% of the letters out of these roughly 16,000 letters, it turns out that everybody has exactly the same letter, So if you're following, that would mean Eve had that letter, right? Now, if we look to the places that had variability, we want to look to the variable places, of course, in the mitochondrial DNA. What we're going to notice is that, let's say everybody in the world has an A, but then one person has a T. Well, logically, what did Eve then have? Well, most likely an A. And we can then apply this logic. And as an outcome, as a result, we understand that we actually have the Eve consensus sequence. And this is the type of information.
We don't as far as I know, Dr. Dan, do we have the Eve consensus sequence? Which
So my knowledge of like molecular clocks and dating is somewhat limited, but I can tell you this much, that from what I recall about from mitochondrial Eve and the and the consensus sequence that we have on her, right? That sequence does not refer to an individual. It refers to a population of individuals that contain that sequence and that pass down that sequence through a specific lineage, right? Because obviously, from an evolutionary point of view, Eve was not the first person that had that sequence, right? It was passed down by other people, but there's limits to how far we can probe that with mitochondrial DNA and in terms of our historical understanding of it.
yeah, it isn't like we have the actual sequence or that even. I don't know. It just doesn't make any sense to me. That part made no sense to me at all. But, you know, coalescent theory, basically they can figure out a time, but let's say, okay, he's describing how to determine a consensus sequence, right? And that kind of gives you a time, but I'm not sure if what he's not saying is techniques that we used to do this return a convergence time of 100, 200,000 years ago. But it isn't like we actually have that sequence. I mean, we can make some guesses, I guess. So if he wants to play that card, that's great. If he accepts that he rejects the associated date, yeah, but that is the card that he plays. So basically, he has the Eve sequence. Okay, so he goes on and with his criticism
that we can then look at to realize that overall, we're only a few mutations removed. Anybody on this planet, only a few mutations removed from the Eve consensus sequence. Extremely low variability in the mitochondrial DNA. Okay, and the critics never address this as to why. Okay, and we're gonna.
Okay, the critics never address this. It's addressed in almost every textbook, I think. I don't know. It's just
never addressed particularly why the dates are.
Why it's conserved. Okay, there are conserved parts of the mitochondrial DNA is highly conserved. Parts of it are not so highly conserved. Most of it is, I believe, and it's because it's very small, very tight. It's got a lot of genes out, a lot of gene density, and I don't know.
Yeah, there's not too much room. Like unlike eukaryotic DNA or nuclear DNA, rather. There's not much of the mitochondrial genome that actually contains that contains non-coding sequences. A lot of it is actually coding regions. So there's not much room for where it can tolerate mutations on in that genome, if I'm not mistaken.
Yeah, and I'm not entirely sure, but I don't think it's just a couple of mutations. I mean, there's quite a bit to work with. So I don't know. I'd have to look at it in detail. And I can't look at everything. So I'll play a little more.
I'm going to go over their arguments later as to some of their rescue devices, but I wanted to make that clear, at least for the audience, okay? But reiterate some of Dan, some of his points here. He's going over the pedigree rate, okay? And the pedigree rate is essentially, okay, is when you look at parents and offspring, and then all you have to do is look at the differences between them. It really is that simple, okay? This is the pedigree rate.
No, it fucking ain't. Because there's a germline mutations or substitution rate, and you cannot, and the substitution rate takes into account populations, genetic drift. It takes into account what else? I lost my train of thought. But I mean, it's a lot deeper than that, and that rate is a hell of a lot lower than the pedigree rate, and it's always going to be. And you can find, I think, pedigree rates where there are zero mutations between the parent and the child. And you can find another one that have 130 mutations in that D loop or some of the non-conserved regions. But it's all hit and miss. But the thing is, all of that stuff levels out. And the only way to find that substitution rate is to make a guess about the amount of time. Now, Dr. Dan, I think originally you were talking about some island or something where we know how long ago the people split off, and we use that time, and we had like a fixed time, and I think we were able to, rather than just using fossil evidence or using guesses as to when we split. I don't know. Yeah, and Standing doesn't deal with it either. So if you go on in this video, he doesn't deal with anything. That's all he does is a sales pitch to his audience. And I wonder if I mean, he's got what? He's got like 600 people that are going to listen to this. Do these people realize that he is lying? Okay, Canary Islands. Okay, so I did not imagine that. Vanuatu. I want to take a look at that study. Anyway, it's just, God, I don't know. What do you do with these people? Lies and more lies, and it just keeps going on. And he doesn't really. I listened to another maybe 5 or 10 minutes of this, and he wasn't addressing anything that they said. Oh my God, do I dare? Well, he's an atheist. He's an atheist, so I could give him a link.
I think we should take a vote.
Oh, Danny boy?
Yeah. Depends if we want to, what topic we want to focus on tonight.
Yeah, well, I can always stick him in the back room if he gets mouthy on us.
Fair enough.
I'm going to be tough. And you, Leo, if you and him get into it, I'm sticking you both in the back room, okay? Leo?
I'll probably just leave.
Is that fair?
Yeah,
you're going to
rage leave.
You're going to rage quit on me?
I don't rage quit, but like if I can't like tell people that they're wrong and show that they're wrong when they're obviously wrong, then I'm just not going to be involved in the discussion.
Well, I'll take you out of the back room one at a time, okay? And let you guys have your,
Just like on Friday, you kept muting me, but I wasn't the one who was like wrong on every fucking thing that I said.
Yeah, but you also were the one that was over talking him all the time too.
Yeah, I was over talking the guy that was over talking literally everybody else in the stream. I was in the wrong though, right?
Well, no, you weren't. It's just that you could have made my job easier because I did not want to mute you. I was fine with it. Yeah.
Notice how I only was acting that way towards one person, Sam.
Okay. Oh yeah.
For that.
Yeah. Well, you guys put me. I mean. You guys obviously have, I'm obviously biased to not muting you. You guys are putting you in the back room because you're on the hyena team, but you make my life difficult. Do you understand how you make my life difficult?
Sometimes, but I wasn't going to let Sam do to anybody what he did to Peter.
What is Leo talking about? He's not talking about the discussion I had with him.
Peter.
Is your name Sam?
Yes, that's my middle name.
But that's not your name.
It is my middle name and name, Leo.
It's not your name.
Leo, are we already starting here?
Oh my God,
Leo, I will put you in your. I'm just kidding.
I will put you in the back room.
When is this? When did this happen?
Friday. Do you know who Redefine Living is?
Yeah.
Oh, okay.
Yeah, you were there.
Oh, wait. I've talked to Redefine Living, and it was a very interesting discussion. We're friends now, but it wasn't because we made any progress in the discussion. But anyway, no, we respect. There's mutual respect. But yeah.
He and I never make progress.
I have respect for him.
I already was like.
Well, he's nice to me. We're Sorry, Leo.
Peter DeTucker from Gonna Go For It. He was having a discussion with him, I think, like sometime last week, and Sam Redefine Living. Apparently, his name is Sam. Was not being nice at all, and I already had it in my mind, like, okay, I know how this guy. I know the game he's trying to play. And I'm not going to let him play it by playing it better than he can. And that's exactly what I did. And I have no regrets. No regrets.
What were y'all arguing? Oh wait, I don't want to. What do y'all talk about? Your Mr. What's it called? Standing for Truth stuff.
Well, Standing was there. Bob Argotha was there.
I like Bob.
Um,
I like Bob.
I
a
like
lot.
Bob. Yeah, I like Bob
Okay,
too.
okay.
Is Bob?
Because he listens to me. Does he listen to y'all?
Yeah,
Okay, yeah.
Bob.
Argotha.
Trying to find the ovals.
Oh, Argotha.
Yeah.
Yeah, that guy's kind of weird, actually, in my opinion. I haven't really interacted with him, but I've seen interactions with people with him.
He's got a very Amish beard, I must say. He does.
Interesting way of characterizing it.
What's that?
That's an interesting way of characterizing it, but that's
He
fair enough.
does. It's
There we go.
an Amish-looking beard.
Ovals.
Yeah,
Boo-boos.
ovals.
Just don't ask him if he's put his fist in horse's ass.
I don't think so.
Then you'll know if he's a mechanic or not.
Should I play this?
Yeah, go right ahead.
No, like, he's basically saying, I don't know. It's one way or the other, but I'm going the other way. Now let's use our brains for a moment and think for a moment. If you were Dr. Nicholson and you were using things to study, and you weren't sure if it was somatic or germline, what would you do? What if there was a very easy way to get a hold of the author of the original paper and ask him? I know. Maybe email him. And spoiler alert, that's exactly what I did. I emailed the lead author of the Bing study, Lynn Bing. I actually emailed them, and they got back to me. And we are going to have a lot of fun with that response later on. So with that, I will turn it back over to you.
Thank you. Yeah, so we're gonna wind up right with that email. So I want to first highlight the other major problems with this paper. So I'm going to go back to my screen here because it gets better. But wait, there's more. What is confusing about that paragraph that David just read is that you have the somatic rate, you have all the mutations of all the cells in the body, the muscles, and the neurons, and the stomach, and everything else, and then you have the germline, the mutations that are in the cells that produce gametes that could.
You should really slow this down.
Now, that
You
sets
have to slow
is
down,
wrong in terms of calculating a long-term.
Why do
mutation rate,
people
because
speed it up
we're
anyway?
doing
Just
a lot
let it
more
play
filtering
at normal
than
speed.
just somatic to germline. So I used these slides when I talked to Sal Cordova a few weeks ago on the same topic. And I'll just briefly go through them here. But if you consider this oval, this is all mutations, every tissue in your body. Now, the germline represents a subset of that. This is not to scale. The germline does not represent most of the mutations. The germline represents a teensy tiny fraction of the mutations in the body, right? Just think about just in terms of like numbers of cells. How many cells are in the ovaries and testes compared to the rest of the body? It's just not that many, right? So the germline mutation rate is going to be a very small subset of the total mutations that occur in the body. Now, Jeanson basically cuts off there and says, but if it's in the germline, you're good, that can persist. No, because you still have to get into the gametes. And this is a problem for both males and females. Not every cell that could make sperm is actually going to ultimately make sperm. Most cells in females in the ovaries don't ultimately become secondary oocytes. Most of them don't. So, like, just think about how the ovarian cycle works. You have a bunch of follicles mature every month, and then only one of them is actually going to go through ovulation, right? Sometimes it's two, and that's why you get fraternal twins. But for the most part, it's one. And then the other, like, 3, 5, 10, 15, however many there are, those just go away. They're dead, right? So all the mutations and the ones that don't mature, those mutations are gone. So you have a germline to gamete filtering step where you lose a bunch of mutations. Now, those gametes, the vast majority of those don't go through fertilization, don't become a zygote, right? So again, you have another layer of filtering. And then the zygote, of course, has to successfully grow into an adult for that mutation to persist in the population. And then the adult also has to go through, in the next generation, you got to go through all these steps again, right? You have to all the mutations in the adult, they have to be in the germline, make it to the gametes, yada, 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 over many generations in order to actually persist in a population. What we care about when it comes to long-term mutation rates, time to most recent common ancestor calculations, what we need to do is use the substitution rate. So here's what Jeanson claims. He says, I use the germline rate, it's cool. He ignores all the levels of filtration kind of below the germline rate. He's just pretending everything that makes it to the germline, all those mutations persist. Okay, David, go ahead.
Yeah, by the way, I did a Command F on my Mac just to make sure I didn't miss anything. The word substitution.
What's happening?
Well, once again, that was every time Dan speaks, he just wastes time going through, germ cell line mutations, talking about the gametes, talking about how somatic cell line mutations don't get passed on, and how we need to
Just
look to the
wasting
long-term
time.
mutation rate, because a lot of these mutations are filtered out, including from generation to generation. A lot of them won't even be passed on, and therefore the substitution rate is more important. I mean, just the same thing over and over again. And yet we point out, yes, we understand all these basics. We understand this.
Then why don't they get it?
Then that raises the serious question, though. If you understand that those things need to be calculated and factored into the overall calculation, why are they not factored in when creationists do their studies, right? Like, why is this being deliberately ignored?
Right.
Because that's the point. It's one thing to be ignorant, right, of the variables that can, kind of obfuscate and muddy up data, so to speak. But it's a different thing entirely to be aware of that and just say, oh, well, fuck it. Well, we don't really care about the muddying. Well, at that point, you're holding yourself to a standard of dishonesty. So I mean, which is it? Is it just ignorant, or do you just not care about the fact that the variables that are being, that are being discussed as relevant aren't making it into the papers of creationist literature? It's either one or the other.
So it's like, okay, every time Erica or Dr. Dan do a comeback video, Standing for Truth gathers his crowd. They get the pitchforks. They light the torches. They go, yeah, yeah, and they go marching down the street. They get to the castle, and then they stop, and they sit around and look at each other's torches, and then they turn around and go home. They never even go in the castle. I mean, and he keeps doing this. What the fuck?
The thing is that's not a response really to what Dr. Dan and what David were saying. It's like, oh god, Dr. Dan keeps raising the same points. It's like,
yeah, okay. because I mean, see what
it's this nasally kind of like childish response, and it's not actually addressing like the substance of what's there. Right? It's not actually providing like a substantive counter to the overall point.
It's more than a pep rally, though, Dr. Dan. They actually get the pitchforks and they dress up and they get the torches lit and they march down to the castle. They just don't go inside the castle. So it's more, it's oh my god, these guys! I can't.
So you're saying they do it medieval style, where they just sit outside the castle and go on a food strike for 2.5 fucking years, hoping the people inside will surrender.
Yeah, maybe that's kind of what he's doing.
By the way, Danny, I wasn't watching anime. I don't watch anime. I was watching the end of Wedding Crashers, which is one of my favorite movies because I fucking love movies.
You don't watch anime?
No.
What a loser.
Well,
God,
hey, I would rather.
Okay. This is one time I agree with Danny on something. God, praise Jesus.
Yeah, praise Jesus. That's what he said.
Who the fuck?
So we got a
Christian
on our hands.
Why are we praising some Mexican guy who does gardening in somebody's backyard?
Praise Jesus.
Do you think anime is like?
My garden, okay,
oh
fuck you.
Why don't you watch anime, Leo? This doesn't make any sense.
The thing that comes remotely close to anime that I watch is the Boondocks.
That's
my
anime.
god! Someone throw him out the window. Someone get it. Put him in the backstage. Speed
I would
give
also
moderation. Moderated powers.
Because I don't like watching mouse moves like
kids,
and then hearing poorly dubbed American voices over it. That's not what I want to watch.
Wait, that's why you don't watch dubbed.
Kids.
But I can't speak Japanese when they're saying, oh my god, I have no fucking clue what they're saying.
So racist.
Okay, the thing is,
Japanese isn't a race. Races don't exist.
Japanese is a race.
No, it's not. It's an ethnicity.
Do you think gay people are a race? I think they're a race too.
I'm about to mute you two. We're moving on. We're talking genetics.
I don't understand this. I think homophobia is just a subset of racism.
It's not.
And then he doesn't stop, and he's headed for the back room. I swear to Christ.
Y'all should take the bait.
You're going to the back room.
Oh, he's a big fan lady.
I prefer most animes. I prefer dub, but some animes I will not watch in dub.
Right,
because
is he going to the back
you
room?
too. Oh, beautiful! I can't put him in the back room because I think he's. Did
he's
you,
part of
did
the
you
wolf
come in
pack.
through? Did you come in through StreamYard Immutable, or are you?
I came in through StreamYard. You removed me. I'm taking the stream down.
God damn it!
Well, he's part of your wolf pack, so it's okay.
Hyena pack.
Fucking Ian.
Not that
You know how
fucking
to read the
complicated.
motorboat? Oh fuck.
Okay, I'm gonna.
Owen Wilson and Vince Vaughn together just crack me the fuck up.
Shall we go further? Afskin.
Afskin. Let's see if standing actually provides it.
Skin.
Okay. The fact is, these things have been considered. Here's a couple phylogenetic trees that give you the bigger picture view. And it doesn't matter, even when you consider African genetic diversity, the human population, African, non-African, only a few mutations removed. That's it. You're never going to get to your evolutionary date. Okay, never. That's why they won't make predictions. So, but at least we're just going through the video minute by minute, so nobody can accuse us of not addressing every single point. But the assumption after assumption being made by Dan.
Are they using? Are they trying to prop up the dates used by? Oh God, what is it? Substitution mutation rates on the D loop.
What's that?
Are they getting their date by substitution rates on the D loop?
I think that, yeah, I think the author that did you listen to this the video that Dr. Dan and David Neff did?
Yes, I did.
I've not got around to it now.
Anyway, they actually contacted the author of I forget which paper it was, but the author came back and said that isn't what they were trying to do. They were trying to they didn't care that it was, they didn't know and didn't care that whether it was somatic or germline. So what they were trying to do was not what Jensen used the information for. That's my best understanding of it. But they get to it later in the video.
I'm asking about, like, Standing just brought up the whole date thing. I'm asking, is the date that they're trying to get to being arrived at by using the D-loop substitution rates?
They use another researcher's paper to do it, and I think he uses D-loop, but I'm not sure. I think he uses D-loop, yeah.
Usually not a good idea. D-loop is highly variable substitution.
Yeah, Dan just posted a paper.
Doesn't usually provide if I'm recalling right, doesn't usually provide too accurate mutation rates.
We got a paper from Soares correcting for purifying selection and improved human mitochondrial molecular clock. Let's see. Full genome. He's using the full genome then. And I forget the paper that was involved. Do we have a link to that original paper that they're talking about?
Do we?
I don't know. I'm hoping Dr. Dan's going to put one up there. Of course, he might have to get out of bed to do that. I don't want him to do that.
And then the waste of time just explaining basic scientific knowledge on pedigrees and substitutions.
Oh my God.
And so on and so forth. So anyways, I'll hand it over to you, Matt. What are your thoughts on everything that was just said by David there in terms of arguments? Because
that always gives me a chuckle when he asks what Raw Matt's thoughts are. That's kind of funny.
That's an interesting question because you never know what you're going to get. It's a lot like opening Schrödinger's box. You get to find out whether the cat was dead or alive all along.
Not if you subscribe to the Everettian model.
Yeah, that's true.
But yeah, Tainted Matthew just shouldn't talk at all.
Yeah, Dan didn't really have any arguments, but what are your thoughts?
Yeah, no, well, Dan just is used to teaching, so I don't even know if he notices it. That's just what comes out of his mouth. He's a teacher, right? So he's just constantly teaching people. He expects them to not know what the hell he's saying.
Hold on.
So I can understand that.
Skin of the fours.
Whoa.
Skin of the fours. Skin of the fours. Thank you.
I don't understand skin of the fours.
Well, that's too goddamn bad. I'm saying the same thing, so get used to it. Anyway, I'm pretty sure Dan is either, what is he? Isn't he an associate professor?
Yeah,
I think that's a little bit different than teacher.
Yeah.
Right.
And I'm not sure what Matt is trying to imply here. Maybe Matt's experience was a little different in the public education sphere. But normally, like, if professors are talking really quickly and like breezing over information really quickly, you can ask them to slow down. You can ask them to kind of like elaborate on points that are confusing. Does he think that like professors are paid to just kind of like risk over information and to not make sense at all? I'm confused now on.
So
This is how you know that none of these guys have ever spent any fucking time in a collegiate setting. And according Dan said, he's an assistant professor, still a professor, which I think is a little bit different than a teacher.
I think Raw Matt is getting his knowledge of professors and their teaching tendencies from probably the Ben Stein's The Movie Expelled, which I still think we should review.
I've never been to like a university, but I've watched enough lectures from Stanford and MIT and have talked to enough of them that I have a pretty solid understanding of the difference between your average teacher and a professor, whether assistant, associate, distinguished, emeritus, doesn't matter.
Yeah, the paper that Dr. Dan linked kind of says that 5 to 6 million years ago for the chimpanzee split is too recent. Slightly too recent, so it's pretty close. I think that's basically the point of the paper. Anyway, you want to listen to Garbage Man talk?
No.
Not really, but let's do it.
See what Raw Matt continues to spew.
Okay, maybe Raw Matt usually doesn't talk too much. We'll see if he ends it.
He brought up saying that you can't take a germline mutation rates back to a most recent common ancestor, but that's done all the time. Here was a study that was done, and she traced using the hypervariable regions 1.
Hold on. Oh, let's see here. Oh, I want to take a little snapshot of this one. I don't really care to get the chick's picture, but okay.
And 2, and she said the most recent common ancestor would be right back. She tested Europeans. Okay, this is just the European lineage. So we would say this would be Japheth's line, Noah's son that spawned the Europeans.
Hold on. Is he saying that Noah is fucking European?
Who is it?
No, Noah, who's from the Middle East. Apparently, some of his descendants just got, inherited some whack-ass form of albinism over the generations and apparently gave rise to the European branch of humans.
I'm fairly certain that Raw Matt is probably a race realist. Like, I feel really, really confident that this guy thinks that, like, black people are just genetically inferior. And I get that vibe every time.
I mean, I can make a joke right now, but
I don't get the sensation that Raw Matt really thinks too much before he talks.
Or at all.
I'll leave it at that.
Oh, he shows a picture of this lady and he gets a quote, but he's not showing the paper.
Well, because when Erica was doing her absolute and utter destruction of Raw Matt last week, she cited a couple things that he said that really, I'm like, it's borderline, like.
Oh my God, we got Dr. Dan out of bed. We did.
Nazism, maybe you could say at best, ethno-nationalism, but I'm just like, yeah, that's something that you hear from neo-Nazis.
I don't know who Raw Matt is, but
good.
He has
Good. Save yourself some brain cells and don't find out.
He's a fruit or a fruitarian rather.
Speak.
But what makes you think that he has ethno-nationalism vibes?
Just the way that he talks about genetics and how it relates to the races and how some of the stuff that he says, at least to me, like, I'm not saying that he is. What I'm saying is it's very esque, if you know what I mean.
Yeah, like the undertone. Their undertone.
Neo-Nazi, yes. That he tries to almost talk in this way that seems like that Europeans are just kind of better than everybody else. And I could be wrong, but like you said, there's some undertones there. That's really, yeah, not at all. Not at all.
Has he used the term Aryan before?
as far as I know, he has not, but a lot of them try not to use that term particularly anymore because obviously it kind of is full. To me, using the term Aryan is mask off neo-Nazism. I mean,
Yeah, I was
Can
kind
we
of
get off this topic?
So this is Evelyne Heyer.
Yeah,
Yeah, that
Rom
topic
is
makes
stupid,
no
but I'd be
difference.
surprised if he'd be that stupid to use that term.
Okay. Hi, Dr. Dan.
Hey, how's everybody doing? I
we
missed
get
a
you-
couple minutes in there.
Did we get you out of bed? Okay.
Yeah,
So,
I was taking notes on some other stuff, and while I was listening, and then it's like, you know what? Why don't I just get up?
Dr. Dan looks like a biologist. I mean, looks like a scientist. Do y'all get a kind like if you look at the all the
Yeah.
philosophers in Yale and Harvard, they're all these old men with really messed up white hair. Like I mean, right? Like the Einsteinian, kind of.
Dennett.
But I don't know. There's a kind of set of phenotypes that is correlation between a particular person and their academic interest.
Okay.
I'm an academic realist, so.
So how do we feel about the non-response from the brain trust?
It blows me away.
I would have expected nothing less from Lügengemeinschaft.
I mean, it's just there's the key thing with like all the filters of why you lose mutations over time from the germline. That's the key thing, and there was no attempt to address that at all. They recognized that what I was saying was correct, and then said, "Yeah, but we know and we deal with it." But like the math doesn't reflect that at all. It's just not there.
I mean, and that's kind of
this is germline, right?
And that's kind of what I was alluding to earlier, the fact that like it's one thing to be ignorant of those factors and say, okay, well then creationist research should update, to contribute the effects of those variables. But it's one thing to be, complicit in defending, to have knowledge that those variables are actually relevant, and then still try and pretend like, but we are accounting for those things. Well, no, you're not.
Right. Show me the math.
Well, the thing is, Dan, I think it was yesterday, I'm pretty sure it was yesterday in the weather report. Standing was in the side chat. Maybe that was today. I don't know. It was one of the two. And you said something like, hey, look, if you think you've got it, just show me the math. And I'm just like, wait, did Dan just ask Standing for Truth to show him the math? You expect Standing to do math?
Hope springs eternal.
I'm still a little bit giddy about the fact that Sal, who's gone through a lot of physics, made a log plot and somehow or another got a linear regression out of that, and I'm like,
yeah, with random numbers, and I've been meaning to look into that for like a week and a half, and I got sidetracked on 6,000 other things. So, but are you familiar with that one, Dan?
Familiar with which one?
Log plots.
Cell did this log plot to prove that
the
DL ratio for amino acids.
Stupid racemization dating.
Yeah, and I had this big falling out with him because I asked him to give me the spreadsheet because I thought he actually had data in there, but no, he didn't have data. He had random numbers, and I simulated it with 5 lines of Python code and got the same plot he did. And I haven't looked into, but the plot he got, he was plotting rate against time. And I don't know what the hell he was doing or what point he was trying to make, but it didn't make any sense.
If you're plotting rate against time, you're getting rate per second per second.
Right, and you're gonna get linear. I don't know. I have to look at it more deeply because logarithms are not my friend.
Dimensionally, you're not gaining any. Unless I'm mistaken on the dimensional analysis of that, you're gaining something akin to like the rate of change of
the acceleration. Yeah.
Yeah, you're getting something like the acceleration of decay of amino acids into their racemized forms.
That makes no sense.
Yeah, you're not getting an actual rate. You're getting the rate of change of the rate.
Well, the reason that he was putting it out there is because it had sort of the same shape as the curve that they showed from some paper. And I started to read the paper, and then I got distracted and haven't. But I will track that down, and I will embarrass him if that's what he's doing.
Wait. You said you're just measuring the rate of change of the rate of change.
Yeah. So it's
Yeah.
acceleration.
So
Hey, guys.
he's essentially just doing because that wouldn't that be a second derivative?
Yeah, you're essentially taking the second derivative of you're figuring out how quickly a rate of change is changing.
Mm-hmm.
Yeah.
Yeah. It's an
Did they
acceleration.
even bother to do the first one?
No, I
My
mean,
question.
I don't know. I mean, the thing is, it's like, why are you? It's very rare in chemistry that you're actually concerned, like, with acceleration of change, unless you're concerned, like, with some kind of fluidics or some kind of process that might refer to, like, reactions.
Yeah, it's going to be pretty specified.
It's pretty specified, but you wouldn't do that if you're trying to figure out how quickly like a peptide degrades and its amino acids become racemized into a proportion of L and D forms.
Mm-hmm.
That like a derivative plot of rate of change isn't usually significant, and it's amusing to say the least.
Yeah. I feel like
let's
Sal
see.
is the one that's trying to take all the creationist talking points and actually make some form, even if half-assed, attempt to like mathematically quantify them. I would just argue that he isn't really being that successful.
So this is the paper that he's showing the lady's picture. Basically, it's Evelyne Heyer, and this is the paper that Matt is showing in the video. And I have no idea what the paper is. First time I've seen this one, but
Yeah.
Hey guys, how you all doing tonight?
Doing good.
good.
Hey,
Yeah.
Dr. Dan. Hey, Dr. Dan, it's good to see you.
I'll open this question to both Walking Fish and
walkie-talkie.
and David Neff.
Hello.
Have you guys ever seen like a rate of change, like that kind of plot before, like a rate of change of what we were discussing, essentially, like the rate of change of rate of
Like
change?
the rate of change, basically.
Yeah.
In chemistry or in anything? Because in physics, it's all over the place.
Chemistry particularly, because this is a phenomenon about like how peptides are degrading and their amino acids are racemizing.
Not off the top of my head.
All right. Hey guys, I saw you guys were talking about the video that's standing for truth day this afternoon about completely destroying me and Dr. Dan. Can someone fill me in on what exactly they said? Because I was busy and I did not get to watch it yet.
Yeah, same.
He didn't seem to say
I caught about
anything.
half of it on and off, and there was nothing serious in it. The parts that I caught were like
Just a bunch of fluff.
Yeah.
I mean, it was their usual stuff where
Yeah, just like completely not relevant to the actual argument that we were putting forth.
There was stuff that was not relevant. There was stuff where like they'd agree with us and then just move on.
Roll on. Yeah.
I like the way that Dan put it in the side chat where he said it was pretty much just like a creationist pep rally. There was no substance whatsoever.
I like my analogy.
Yeah, it was. It is not worth a response video. It's just like there's nothing there. We made arguments. The arguments were quantitative, like there's math, and they're saying no, those have been dealt with. Okay, do you have math? If not, then you didn't do anything.
Yeah. By the way, Dr. Dan, do you know if they ever spoke about what Ding actually said in the email?
They basically cut that off. They actually kind of stopped before the end of the video.
Oh, well, of course they did. And did they ever address the fact that his sample size was too small?
I know they got past that part, but I didn't see the part where they talked about it, like where the part where actually I did see the part where he said where you talked about they didn't use African lineages, and Ramat's response was, well, of course he didn't use African lineages. We don't have any data on that. And I'm thinking, here I am thinking this guy is a scientist. Like maybe he would go out and collect the data or something. Yeah. Not just stop. But that was Ramat's excuse that no one's collected that data yet, so you can't have expected him to do that.
Yeah, well, he had it for 5 years.
Oh wow,
It
full
was
house
it was
tonight.
not. There was
Yeah,
nothing,
full
there was
house.
nothing new. There was nothing insightful. There was nothing quantitative, which is what I'm looking for. I want to see the math. If Jensen, they said that Jensen accounted for things like selection and drift and all that stuff. I
Math
want
doesn't
to see the
exist.
math on that.
What do you think Jensen would send us his spreadsheets?
Jensen is notoriously not open to responding to people, and the
Responding
supplementals
to critics.
for the supplementals for his blog post is almost non-existent. It's one half-ass spreadsheet that doesn't have enough in there. I tried to like reverse engineer his calculations one time, and you could kind of do it, but it doesn't have the raw data, so it's not easy to do.
Yeah, well, I sent him an email and I asked him for the data on his. I think it was it was for that little tree he does, the mitochondrial tree.
Wait, do you have an email for Jensen himself?
I do.
Oh, can you send me that?
I can.
Email me that. If you don't mind, send me that email address.
Anyway, so he responded to me with a link pointing to something about Africans having babies when they were young or something.
He requires a 15-year average generation time, not minimum.
Yeah. That's what and that wasn't what I asked for. And then I gave up on him. That was about a year ago that I did.
Mm-hmm. Yeah.
I should not have given up on him, but maybe you'll get further with him. I would love to see his actual.
I'm gonna ask him.
Yeah.
I'm gonna email AIG. I'm gonna email him directly. Like I'm gonna ask him this stuff
By the
because.
way, Dr. Dan, I've actually been starting to put together a paper that I want to try and get published in the Answers Research Journal about actually see if they would actually publish my rebuttal to the paper.
Go for it.
I don't know if that's something you would be that you would be interested in doing with me. We can work on it together, put both of our names on the paper, and see if they'll actually do anything about it.
Say, David, when you get the rejection letters, please keep them and show them.
Yeah, that'll be fun to see if they actually publish something that is substantial of a rebuttal to his work.
I mean,
so Dr. Dan, is that something you'd be interested in?
I'm not interested in playing an AIG sandbox because they want to have it both ways. So AIG, they have their Answers Research Journal, which they claim is a peer-reviewed journal. Except it's not. It's a blog. It's an in-house blog is what it is. But they want to pretend that they're doing real science and publishing it on their blog. Now, if it was a real journal, I'd say, sure, fine, we can have this scientific disagreement in a scientific journal. They will not put their work out there through the real peer review process.
Oh, of course not.
So nobody else, no real scientist should feel obligated to respond through that process to their work. They are putting their work out through, informal, non-peer reviewed channels. And to me, then that means that those same types of channels are sufficient to respond to them. So, Reddit posts and YouTube videos is the appropriate way to respond to that. Or if I have a blog, I can put on the blog. Like pretending that the Answers Research Journal is an actual peer-reviewed journal is giving them too much credit. It's not. And what they want to do is be able to claim that it's peer-reviewed, but use it in such a way that they can control it. Right,
exactly.
If you read the fine print of like emailing them stuff and submitting stuff to them, they can change it around, they can edit it. They don't have to publish the whole thing. They can publish part of it. It's their set of rules, but they also want to claim that it's a legit research publication. Can't have it both ways. So no, if you want to submit to it, I'd be happy to look it over, edit it, whatever. But like AIG can if they want a response from scientists, they can
actually put it in a real journal
and actually publish in a real journal.
Yeah. By the way, this is the thing that I don't understand about Dr. Jensen is that if he actually really wanted to calculate and overturn the scientific consensus regarding the time to mitochondrial Eve, he would have put that stuff in a real journal like Nature or Science.
Mm-hmm.
Yeah.
And I love how I've heard one part where saying Richard said that we should put it in the Answers Research Journal. Then if that's the case, then they need to get off YouTube and start publishing their stuff in an actual peer-reviewed journal and stop writing books on Amazon. That being said, yeah, I've been looking at some of the stuff that Fraud Matt has been writing, and it is just atrocious. Like, how did he pass high school writing?
I don't know.
I mean, you're assuming he did. Oh,
The pull of praise. Oh,
Leo, my eye, my ears. Jesus Christ.
Let's see.
I don't praise this channel very often.
Dan, Andrew has a question for you. If are you ready, Andrew?
Oh yeah. Are we done with genes or?
Are we? Well, I mean, we can ask a question. It's,
Oh,
Go for
okay.
it. I'm gonna
I
answer
just
for a few
want to.
minutes, and then I'm gonna go to bed for real. I swear.
Okay.
okay.
I just wanted to ask something real quick to you, Dr. Dan, before we continue with the discussion and before you have to leave, if that's okay.
Yeah, go for it.
So you probably know that I had a discussion with SFT about nested hierarchies, and so one I guess well we've already talked about how phylogenetics does not assume common ancestry a priori. It makes predictions, right? So I want to ask if he like he keeps using the model of cars, for instance, supposedly falling into a nested hierarchy. And so I want to ask, for instance, if we know that if we use cladistic methods on something that actually has a shared history of ancestry, like living things, then we get a consistent, unique nested hierarchy. And I want to ask, what do you think we would get if we applied those same cladistic methods to something without knowing if it had a history, such as like designed objects or something like that? What pattern might we see?
Well, I can tell you because we've tried to do this with viruses, because viruses are polyphyletic. Unlike cellular life, viruses don't share a common ancestry. There's multiple independent origins for viruses. So you can do phylogenies for groups of viruses, but you can't link all of them. And basically what happens when you try to do the phylogenetics across groups that are unrelated, that are so different from each other, you lose the phylogenetic signal. So you get a series of radically different trees, radically different phylogenetic trees that are all equally likely. And what that tells you is we can't say anything about this relationship. Does that mean for sure that there's no common ancestry there? No. But does it mean that we can figure out what the common ancestry is? No. And that's the difference, where if you get a clear relationship, that tells you common ancestry. If there's no common ancestry, you don't get a clear relationship.
Yeah.
The reason you could do this with things like cars and stuff, if you were to somehow tally up a matrix with like cars, is that it's basically convergence, right? Where like, yeah, they're different, Toyotas and Fords and whatever, they're all different, but they all kind of work the same way and do the same thing. But they don't evolve, right? We've designed them. With living things, they evolve because they propagate, right? That's just, if you propagate, you have offspring. The generation over generation, there's slight differences. And we can do short-term experiments and document and actually test those techniques. And there's one of my favorite papers from 1991 is actually a test of this, where they took viral populations and split them iteratively, and then just sampled the end results and then reconstructed the phylogenies using a bunch of different techniques, basically to validate the techniques. And when you do the same thing for groups that are not related to each other, it doesn't work, because it's all phylogenies are probabilistic, right? It's degrees of certainty, and it breaks down if there isn't actually a relationship there, because you get a bunch of potential answers to the question that are all equally likely but are all contradictory with each other. And when that happens, that tells you no common ancestry. And so we make these predictions, and it's not just that the structure resolves, but it's that when you take subsets of those data sets, it should resolve. Or when you take a different data set, it should resolve the same. Or when you look at components of that data set, you should see that nested hierarchical pattern. So for example, in things like endogenous retroviruses, where there isn't a functional significance, but we know they're there. Like, why are they degrading across different genomes in a time-sensitive way if there isn't actually common ancestry there? Why do we share more in common with things that we fall closer to phylogenetically than things that we're further from phylogenetically? So basically, you're using the initial data set to make that tree, and then based on that tree, you make a bunch of predictions, and then you can subsequently test those predictions, and then, as necessary, revise that phylogenetic structure. So people like to throw around, you hear like, oh, there's just lines on a piece of paper, or oh, there's deep time assumptions, there's assuming common ancestry. No. What we're doing is we're saying, here's our hypothesis, common ancestry with this specific pattern. And then by doing the phylogenetics, we are testing that hypothesis. And if we get results that match the basis for our hypothesis, right, then that's evidence that we're correct.
Okay. Yeah. So just to clarify, so you're saying that the difference is essentially that one, cars, designed objects, there's no statistical significance of one favoring one tree over the other if like they're all equally likely, whereas you generally find a high statistical significance for living organisms because they do share a history.
It doesn't make sense to do it for non-living things because the tree implies like inherent offspring relationships. Like it implies vertical transfer of genetic information. Cars don't reproduce.
Yeah. By the way, real quick, can I share something real quick?
Sure. You can. So I really want to do this for vehicles. I would love to, come up with some traits of vehicles. And I mean, even military vehicles and trucks and, those big 300-ton trucks they have up north, and kind of do a phylogenetic analysis of vehicles based on some traits. I think it'd be quite a task, but it might be fun. I'd like to see how it comes out.
Yeah, you could definitely do that in phylogenetic software if you have like discrete characters that you're measuring.
Yeah. By the way, guys, I really like this image. I really like using this analogy of language. We can do the same type of nested hierarchy in phylogenetic analysis on language to see common divergence and how they descended. And we can see this is the same thing that we would see in evolution. And really it's almost the same exact thing because there's descent modification. We modify the pronunciation and meaning of words. And over time you get brand new languages. So again, this is the analogy that I like to use because we can objectively see it and we can do we see the same exact nested hierarchy that we would see in evolution.
Exactly.
Yeah.
Language is a beautiful analogy.
Yep. Good.
Yeah, I wanted to ask Dr. Dan a question if it's okay with him or if he's just trying to sleep.
Go for it.
Yeah, but the question essentially pertains to mutation rates. Like with mitochondria, for instance, because their genome is circular, and most bacteria, for that matter, also circular genome. Does the mutation rate consider... do the mutation rates that we measure take into consideration the, if you will, the topography of the DNA? So,
when you say topography...
So, being that, for instance, like a nuclear DNA, for instance, is very convoluted in its structure, right? It's wrapped within chromatin, and it goes all over the place in terms of how it's being folded. It's such that you wouldn't always expect a particular segment of DNA to be equally likely to mutate it based upon how it's being packaged. Whereas in something like mitochondrial DNA, for instance, mostly, to my knowledge, it's all it's code, right? It's circular DNA. Mitochondria don't bother to pack around.
Yeah, they do have some kind of like supercoiling, but I don't know the degree of secondary structure in there.
Right.
I would expect, we know that there are regions that are more variable or not, but it's unclear if that's variation in per base, per replication mutation rate, or if it's selection, right?
Okay.
So like the D loop is hypervariable. I would be surprised if there was actually a higher, because the units for mutation rate are changes per site per replication. I would be surprised if the D loop had more changes per replication, like than the rest of it.
Yeah.
So it's probably just differences in selection acting on different regions. Yeah, that's my guess.
Okay. Yeah, I was wondering if the relative shape of the DNA kind of close to the site you're analyzing would be a significant type of consideration factor in ascertaining the mutation rate of some segment of interest for trying to measure mutation rate or substitution rate.
Right, well, so that's the thing. So the units are really important here. So mutation rate is mutations per site per replication.
Okay.
Substitution rate is fixed changes per site per year. And the per year is important because it removes variation in generation time from the equation, which means you can do apples to apples comparisons with like humans and E. coli in terms of substitution rate because it's per year.
And it's also the fixed mutations, right? So it's not just any mutation that comes through. It's specifically the ones that are fixed in the population.
The ones that are fixed in the population, exactly.
Yeah.
And going back to what we were talking about before, that's what you use to do your most recent common ancestor calculations, your time to most recent common ancestor calculations, because any mutations that are not fixed may not be there in a year, in a decade, in 1,000 years, whatever. Those mutations could potentially be weeded out, and the vast majority are. So if you're looking at what mutations within, a pedigree, mom to daughter, right? You look at the differences between the two of them, putting aside the problem with somatic mutations, putting aside the problem with the somatic to germline bottleneck, the germline to gamete to zygote, put aside all those problems.
Yeah.
Put all that aside. If it makes it into the next generation, it's still only present. That one mutation is still present at a teensy tiny fraction of the population. The likelihood that it's still there in 10 generations is quite low. So that substitution rate ends up really, really low compared to the mutation rate. And on the Evograd's part debunking of Jeanson's work, he shows a really nice figure. It's in part 6, I think. He shows a really nice figure that shows this, where it's the x-axis, it's basically the probability of the mutation persisting. And on the y-axis is time from present. And it starts off really high, right? Because, if you make it to the next generation, congratulations, you made it. The likelihood that you're there is pretty high. But generation after generation, the likelihood that you persist gets lower and lower and lower, and it asymptotically approaches the long-term substitution rate. So you have to use that long-term.
It actually does make me think of a different type of question, too. And this question is kind of weird in the sense that, does it ever happen that potentially beneficial mutations get weeded out because essentially they're being expressed, or the way they're being expressed just simply isn't sufficient in comparison to other genes trying to
or even simpler than that, just drift. I mean, drift is something that's like, the example I like to use is mice on a barrier island, and depending on where they live, different fur colors are going to be more or less beneficial. If you live in the sandy soil, then being more gray is going to be better. If you live in darker soil, then being brown is going to be better in terms of birds seeing you. If there's a flood on that barrier island, it doesn't matter if you just evolved a much better pigment. The flood doesn't care. That's random, right? So it could be the best camouflage those mice have ever found, but it may not persist just because of bad luck. That happens all the time.
Yeah. That's right. The cruel fate of nature fighting against
and that's another mechanism of weeding out that Jeanson didn't account for, right? You just randomly lose allelic diversity in a population because of genetic drift. There's nothing in there. He just assumes basically that the germline, as long as it's in the germline, then it's good forever in that population. That is a bonkers assumption.
No, I mean, because the way I view it, at least, and correct me if my perception is skew, is that when you're talking about like selection and how likely it is for something to be passed on, it depends on essentially trying to separate, the fitness factor from the environment is really a pointless game, in the sense that if the environment changes, it'll likely influence how it could potentially influence the fitness of the organism to pass down to the next generation.
Oh yeah.
And those changes can be quite rapid, like you said, like a flash flood or,
And this
sudden
is, yeah,
environment or something like that.
it's got surprise. It's complicated. Yeah.
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 I am going to go to bed for real now, everybody.
I think on
entertaining our adult questions.
yeah,
I'm always happy to talk bio. It's what I
Dan,
do.
I think on February 23rd there might be some interesting things said about
Yes.
all that. So anyway,
Wait, February 23rd. Interesting things. What?
about fitness and how things end up in populations and accidents and many
Yeah,
other
that's
things.
we're gonna do that.
Battle
Right?
of the Dans.
Yeah. By the way, Dr. Dan, real quick.
And that'll be
You know, what is the average age of someone starting a PhD program?
Oh, I don't know.
26.
I don't know.
It's about 32, 33.
Average age of starting? No.
I feel like it's smaller for, or like it's younger
It's actually
for
most.
sciences.
I should say that most PhD students are in their early 30s.
Everyone knows that you digest your brain at 30. So
Well,
Yeah,
okay, so
so
it
I was
depends
gonna
on if you
just-
do it right after undergrad or after a lot of people get a master's first. So if it's after undergrad, then it's 22. If it's
Yeah.
after a master's, then like 25, 26.
Right, because I'm 25 and I'm just worried because I'm just starting out at UMBC in the fall. I'm just worried that I might be too old to really get a PhD or anything like that.
No,
There is no such thing as
No,
too
yeah.
old for a PhD.
Yeah,
There are
that's
people
correct.
in their 60s that end up in graduate programs.
Wait, I just looked it up. It says before they get the PhD, they start around age 33. That's according to CBS News.
Yeah. Wow,
By the
that's
way,
late.
Dr. Dan, how old were you when you got the PhD, if you don't mind asking?
I was 28.
Okay.
I was
So,
I went right from, so I didn't do a master's though, so I went right from undergraduate. So I started right after undergrad, and then it took 6 years. So yeah,
Yeah, because what I'm planning on doing is get my bachelor's degree from University of Maryland, Baltimore County, and then maybe do a PhD from UMD, University of Maryland.
nice. Yeah, go for it. Yeah, you're never too
Do you
old.
know
Just
why?
go for
What are there too many variables to decide why someone finishes a PhD in 3 years versus like 8 years?
Yeah.
Like, is it
There are
one?
too many
Okay, it's not
variables.
going to be one particular reason then, right?
Wait,
No.
when you say finish a PhD in 8 years, I'm assuming you mean like going through the requisite degree programs before that.
All right, I'm gonna go to
No,
bed.
not
everybody.
even.
Good night.
So you're
Yeah.
talking somebody that got a
Yeah.
master's and then takes 8 years to get their
Yeah.
PhD? Like I've never heard of that in my life.
No,
I've
I've definitely heard of that.
I've heard of that many times.
Well, it certainly doesn't happen in the physical sciences.
Well, so
It's university.
Hold on. David, you're doing this video in 2 days?
Yeah, I'm going to work one Friday 27th. So yeah, Wednesday. I plan on doing it Wednesday. Though I might change depending on work schedule and all that. So just keep an eye out.
Awesome. Okay.
Yeah, typically, like at least with the university I went to as an undergrad, typically they cap you out if you've gone over. Sometimes it depends on the program, really, but it'll be usually 6 years is the maximum allotted time you can do for a PhD.
I don't know how it would take somebody that fucking long. I don't get it.
So,
I don't
so
get
it
it.
depends on what you're getting your PhD in as well. Like,
It
so
depends on the scope of your study too. Like you can do a lot in 6 years if you're really pushing to the grind and based on the type of thing you want to do. It's usually rare because that's like it's quite extensive. It's quite extensive research that you have to be doing to go 8 years on.
Mhm.
And at that
According
point,
to the CBS News that I just cited, it's the average time to get a PhD, according to this resource, is 8 years.
I think they're talking total time, like the time from when
from
university to get your PhD. Because I gotta say, most places I know of, from Harvard and Cambridge and Oxford, Stanford, Yale, Cornell, places that I've looked at, it's anywhere between 2 and 4 years to get a PhD, depending on what your subdiscipline
The language is not
the
clear
research
here.
that you're
It's
doing.
I'll repeat word for word. It says the average student takes 8.2 years to slog through a PhD program and is 33 years old before earning the top diploma.
I would have to say that's probably the full time, because I don't know anybody that's going to spend 4 to 6 years to get a master's and then another 8 years to get a PhD on top of that. That's an insane amount of time.
I don't know.
Master's programs typically take two years typically.
Yeah, like once you've got your bachelor's, 2 years for a master's, and then usually 2, maybe 3 years on top of that for a PhD.
And even within fields, it can vary quite widely. Like I know people on the computational side of biology usually can get through their PhD programs relatively fast. But people who are on like the ecology and evolution, they actually have to go out into the field and collect data and all that kind of stuff. And that takes up a lot more time.
Yeah. Anyway, guys, it's 11:15. I'm heading off to bed. Good night, guys.
Okay, hey, David.
Yeah.
David, when you have something like that coming up and you're on my channel here, please plug it for us so that we know to watch. Okay.
Yeah, definitely.
Okay.
I actually did a show with Dapper Dino on my channel a few days ago.
Okay.
So check that out.
Did anybody see the show that
Peace out, everyone.
that Dapper had with Otangelo on?
Oh, the falsifications one?
Yeah. Oh my God. They just Dapper and what was the other guy? I can't even remember now.
It was Josh.
Yeah,
right?
I can't remember. Yeah, I think it was Josh, but I can't remember what his screen name. Sphincter of Doom. That's what it was.
Yep,
Screen name.
that's it.
They just bodied him again.
I mean, it's kind of like beating up a disabled child. Honestly, that's
They were talking about how, okay, so if this is your model and your model predicts this, yes, and we can show that this is actually not something that happens in reality, all right, then that means that we falsified your model. Yes, that's what that would mean. All right, but we know this doesn't happen. Well, no, that's not my model. And Erica pointed out, Guts Gibbon in the chat, she's like, it's like the but it's not my wallet from SpongeBob. And I literally about fucking died of laughing. I was like, oh my God. Because that's literally what it was. Oh, it was fucking hilarious. I'm just like, this guy really just does not understand science, which is not something that should be new information to anybody in the stream. Or listen again.
Angela.
Odie.
Oof.
It's the Grasso show once again.
Praise is doing an atheist beatdown. It's probably going to cure me of atheism if I go over there.
What, right now he is?
Right now?
Yeah.
Oh, no.
Yeah.
Is he beating down the atheists?
He's beating down atheists. He does that every now and then. He beats us down and we rise up again. Rise up again.
Wait, can I prevent that from happening by asking Leo a question?
Maybe.
Okay. Hey Leo, how are you doing on QM recently?
On
QAnon?
what?
On QM, quantum mechanics.
Oh,
that's what I call quantum mechanics.
I'm slowly drifting closer and closer to Everettian mechanics, quite frankly.
Oh, okay.
I've been watching a lot of lectures from Sean Carroll, who's like its primary defender at this point in time. So.
Right. May I ask a question about the different formulations of quantum mechanics again?
Mm-hmm.
Well, there's one formulation of it, but mostly it differs in interpretation of how that.
Yeah. That's why I prefer the word foundation.
Okay. So I see in most classes or videos or whatnot, that the quantum foundation that seems to be used the most is that of Schrodinger and the wave function. And I want to know why. Say that again.
Copenhagen interpretation. Niels Bohr and was it Bohr and Schrodinger? Or was it?
Yeah, Erwin Schrodinger.
That's what I thought.
Right. Well,
I'm textbook quantum mechanics, because that's what any textbook you read. That's the foundation that they're teaching from. And if you go to university, that's what you're going to learn.
Right. Well, I guess to be more specific, I'm kind of talking not in terms of interpretations, but in terms of, say, wave mechanics in comparison to operators, which I don't understand, but I kind of want to ask how why wave mechanics seems to be the most prevalent in comparison to like at least in classes, in comparison to like matrix mechanics. By
Well, the thing is
Heisenberg.
that they're is the word I'm looking for, complementary of each other. You learn both, so there's a lot of matrices involved.
Okay.
From what I've seen, you'll see that more. I mean, you see it in basic quantum mechanics, but you'll see matrices appearing more once you start delving into quantum field theory. That's when you I mean, and wave mechanics is still prevalent there, but you tend to see a lot more matrices involved because you tend to work more with groups in quantum field theory than you do in just, one-dimensional quantum mechanics, which is generally what most undergrads are going to learn. If they take a quantum mechanics 2 class that's undergrad, that's when you're going to start getting into a 3-dimensional, maybe 4-dimensional quantum mechanics. But once you start getting into the advanced quantum mechanics and quantum field theory, you start dealing with matrices because you're dealing more with groups than you would in more basic classes.
Okay. Can I ask a follow-up question then?
Go ahead.
So based on wave mechanics and the wave function, do you know like how to explain why quantization of energy results from imagining like a particle in a box? I believe I'm kind of having trouble understanding that.
Yeah, no, I think Immutable would probably be more apt to answer that question since he's actually worked through those problems.
Jesus. Just restate the question again, because I'm trying to figure out.
Right. Well, like I often hear it in terms of a particle in a box where the box has infinite walls in terms of repulsion, and that somehow leads to quantization of energy, like Max Planck saw. And I'm trying to figure out why that is in terms of the standard quantum mechanics.
Yeah. So the basic principle with the particle in a box thought experiment is that that at some point in time you have to at some point, if you consider some interval, at some point on that interval besides the endpoints, that if the essentially that the position at some point in time has to be 0. And the only consistent behavior, there's only 2 options mathematically for what that can be. It can be that this function is that this function happens to be a constant function, which doesn't make sense, because if it's constant throughout all points if it's constant throughout the interval of the box, then there's no point outside of the on the interval it's defined where it would be 0 if it's constant. So it's a trivial solution. But the other one is that you have something that represents a sinusoid wave. Right. And that works for the reason that the sine and cosine functions are defined in such a way that they're periodic. So based on the I believe it's based upon one of the factors of the sine function, I believe it's the periodicity, or there's a technical name for it mathematically. It essentially will tell you how many times that the sine of x or the cosine of x or whatever trigonometric function you're taking is essentially crossing the point 0, because sine oscillates from 0 to, its high points are 1 and -1.
Right.
And so if you change how often sine oscillates, right, there's certain points you can identify with that to be, okay, well, sine of pi is 0 and sine of 3 pi is 0 and yada yada yada. And for that reason, right, because sine can take on values of 0 at various points in time, and even more, especially if you change kind of this periodicity factor to it. It gives you wave behavior,
right?
And that's kind of the way it gets rationalized.
Are you talking about the frequency of the sine wave?
Yeah, precisely, frequency. That's.
I'm going to share my screen really quick, Speed, because I think, and I refer to this animation, I guess you could say, frequently, because I think it really helps visualize this there's a way of describing it, but I can do that at the same time.
Could you link it in chat as well?
Well, yes, but after I'm done, because I have to go back to StreamYard to share it. So let me know when you've got the screen up.
Sorry, Speed. I turned this into a QM discussion again.
It's up. No, that's this goes wherever it goes.
Can you guys see my cursor too, or does that not appear?
I can get on the box.
Okay, so if you can you
Oh,
make
this
out,
is from the wiki, right?
yeah, can you make out the letters at the top?
Yes.
Yeah.
A, B, C, D. So
okay. Is this one of those eye tests?
No, in B, what you have, so you just have right there, you could say it's sort of like one wave in a sense, because there's one arc there. Here you've got 2, now you've got 3. So the quantization, so to speak, is it's kind of like with a guitar string. You can pluck the string, you can pluck it hard, you can pluck it soft, which will give it different quote unquote energy levels, but it's only going to be able to vibrate, so to speak, quote unquote. I mean, obviously with wave functions, they don't actually vibrate, but a guitar string does, in a set of modes. So B would be a mode, C would be a mode, D would be a mode, E and F are modes. They're different modes, but you can't have anything in between those. It's only those modes. It's one or the other. There is no in between. So it's discontinuous in that sense, that you only have a certain set of modes that can be occupied and nothing else. It's either those modes or it's nothing. There is no in between, and so that is
it has to be a whole number, basically.
In a sense, because in quantum mechanics, working with whole numbers is a little
right. I should say it's a whole number of the frequency, perhaps.
I don't know.
Okay.
I'm just more used to dealing with complex numbers, because the wave function is complex valued.
Right. Yeah, I was just asking because I already told you guys I got a new book on quantum mechanics recently called Beyond Weird by Philip Ball. And he mentions this, and I was having some trouble understanding of it, but now visualizing it, I think it makes more sense. And like I know that I shouldn't probably be visualizing it in terms of a wave going up and down, but that does help. Thank you.
Yeah, wave functions are just very statistical, which to me it's the stochasticity of quantum mechanics that makes it non-intuitive, not the math. The math is, in some senses, easier than in many aspects of classical mechanics. It's just quantum mechanics is much more non-intuitive than classical mechanics is. So I mean, you can work through the math, but you're like, I don't really know what this means.
Right. Like, can I give an example?
Yeah, go ahead.
Well, like another example I've heard of is like superposition, which we often talk about in QM, where you have the position can be described. I believe it's a linear superposition of states.
Yes. You know what vectors are, I assume.
Right. Yeah. I think you
know what a basis vector is.
Can you remind me?
Yes, I'm going to share my screen because this is going to make it really, really easy. Just give me a second here.
I guess while you're pulling that up, I guess what I want to say is that if you have this state, I'm just going to speak loosely here. If you have this state plus this kind of contradictory state plus this contradictory state, etc., as a solution to the wave function, how exactly do you interpret that when it's describing actual states of the particle or the quantum object, I guess I should say. Like, I feel like that's an example of what you were talking about, right?
So let me know when it's pulled up.
Oh, I'm not paying attention.
Of course you're not.
It's pulled up
already. Let me go to white. So I'm gonna do 2 dimensions just because it makes it easier than trying to draw it in 3 dimensions. So you have 1 dimension and 1 dimension, and these are vectors. You can write the vector that represents your wave function as, and we'll just go 1 and 2, and I'm drawing with my finger, so ignore the crudeness of what I'm writing. So the wave function, let me do that in a different color. The wave function would be said to be a linear combination of these two basis vectors here and here. So that's The basis vectors are the vectors that essentially define the vector space that you're working on, hence the term basis vectors. And as you can see, I mean, it's a linear combination of both of them. So, and then wave function collapse would be taking this, your psi vector, your wave function, and projecting it onto one of the, in this case, two basis vectors, but in a Hilbert space, they can be infinite dimensional. So you could have potentially an infinite number of basis vectors. Oftentimes you utilize an operator, which I think generally is represented as O hat, or it would really be like the Hamiltonian H hat. Would be your most common operator. That I believe the operator is generally what projects your psi vector onto one of the basis vectors. And then once it's been projected, you would have this vector, but you have to scale it to match the basis vector, which gives you, and I'm just going to draw it here, don't worry about the angle, a new vector with lambda representing the amount by which that was scaled. And this new vector, which is called an eigenvector, which is a vector that only changes by a scalar amount. That new vector, or rather lambda, the eigenvalue of that vector, is what corresponds to the observable. So that would be essentially, so when we talk about the quantity that is measured, what they're referring to is lambda. They're referring to the eigenvalue of that new eigenvector. And so the new eigenvector represents the observable of the quantum state.
So a quick question about that one. The vector you're projecting onto your basis vector, isn't that always orthogonal to the basis vector?
I think so.
Because that's the only way.
I either want to say orthogonal. I don't think it's orthonormal. I think you're right. I think it's orthogonal.
Yeah, I was curious about that because I'm thinking in terms of the matrix mechanics, I don't think you get one result unless you have it being orthogonal.
Yeah.
Oh, by the way, Leo, did you put that animation in the public chat?
No, I didn't. Let me see if I can't find that here. I don't even know what I typed in to get that. What did I type in to get that? I have no fucking clue. Let me just go to my history and see.
UM is fun but bizarre.
Yes, that is very much true. Okay. I'm going to post it in both the private chat and the YouTube chat.
It's amazing just how much of, like, even professional physicists will say this too, is that how much of the essentially how much of the foundations of quantum mechanics are kind of unexplained.
I posted that in the wrong YouTube chat. I was on Praise's stream. Whoops! Let me go back to
really?
our stream. Well, I don't have a wrench, so I don't think that comment would have showed up.
I was gonna say, well, he can probably still see it through StreamYard. So
yeah,
I bet
he's probably looking
pretty
like,
confused.
what the fuck is Leo posting here? What's going on?
What is this GIF? What?
But yes, I do believe that
you can now learn about QM.
I do believe that your state vector, which is the vector representing the wave function, has to be orthogonal to the basis vectors.
Find it.
And it is kind of curious just how much the foundations, if you will, of quantum mechanics are kind of still a little fuzzy. How we don't fully understand them.
Yeah.
Kind of the reason this discrepancy exists and how to interpret quantum mechanics.
Mm-hmm. Now I gotta look at what orthonormal means. I never can fucking remember what that word means.
Orthonormal.
I know what orthogonal is. Essentially,
means
at a right angle to, but
I think the basis vectors are.
Orthonormal is when it's both orthogonal and normalized. So yes, it's orthogonal, but I believe to actually get a result from the state vector, it has to be orthonormal. It has to be orthogonal and normalized.
So they're all ones, right?
Yes.
Yeah.
So that all the probabilities add up to 1, because if they add up to less than that, there's something that you're missing. If they add up to more than that, you probably added too much. Or wait, did I get that backwards? I think if it's less, then you put too much in the two. People can figure it out. If I can figure it out.
Bill Morgan has a premiere on how to deal with depression, stress, and overcome. I bet it has something to do with Jesus.
I would be surprised if it did not.
What the fuck did I just do to my computer? There we go.
Yeah, like I said, I noticed. Oh god, there's shit in my throat.
Well, you know those crackers they give you in Catholic churches? If you snort those, they actually work as an antidepressant.
Oh yeah. We got little tiny pieces of bread.
Oh yeah, you just grind them up real fine and snort them, and
What?
a fucking amazing antidepressant.
But
What are we talking about now?
No, not for you, Andrew. You're a minor. I'm sorry. This channel does not. This channel does not advocate snorting Jesus for minors.
Then why'd you bring that up?
Andrew, rebel against the authorities.
I'm sorry, Andrew. I keep thinking you're a lot older than you are.
Well, with his intelligence, you'd think so.
Well, remember, we're past hyenas, right? This kind of is apt for the description. I kind of feel bad if I wasn't playing devil's advocate here.
Right.
It took me 13.8 billion years to get to this point, so I guess I could say I'm just as old as all of you.
What is the most basic definition of a machine?
I don't know. I'm not an engineer. I'm a theoretical physicist.
I can tell you that I'm thinking about thermodynamically. I would say it probably has something to do with the capacity of an object to perform work on another object.
Using or applying mechanical power and having several parts, each with a definite function, and together performing a particular task. And that's why the cell is not a machine.
Yes, that is exactly why the cell is not a machine.
Well, the thing is, like the most simple way you can argue against it not being a machine in any kind of like conventional sense is that the cell is a very dynamic type of thing. It definitely is definable in terms of the laws of nature, but it exceeds that in the sense that
Nicholson talks about they're self-organizing and they are self-assembling. So they are both, and it's that self-organization that is the most stunning feature of life. That if you change it or perturb it, it goes back to some state, basically.
Yeah. One of the really surprising aspects of life that's very underlooked, or very overlooked rather, is the ability of life to kind of maintain homeostasis, and especially in the way that you don't really think about it too much, but the cell keeps two different environments, right? It maintains its own physiological conditions, and it also governs kind of what types of things that it's going to allow in and allow out. In a very loose sense. It doesn't perfectly do that because otherwise antibiotics wouldn't work. But yeah, cells have a very innate ability to adapt to things around them and to kind of reprogram themselves based upon types of stressors and events that they encounter in their day-to-day kind of existence.
Yeah, he talks about the centromere, and the centromere is like this amazing self-organizing bunch of molecules. It just self-organizes, and then you perturb it a little bit, and it'll, it keeps changing through these states, and that's how the cell divides. And it's this really an interesting paper. I'm about halfway through it, Nicholson, a little more than half, maybe. But at some point, Scott and I are going to go through this. Scott Duke.
Yeah, I mean, the really fascinating aspect of life as well, and the most curious thing about like embryological development. I mean, there's a lot of it, but one of the more basic elements of the fact that there's nothing whispering to a cell how it's to a sperm cell or to a newly fertilized cell in regards to how it's going to divide and how it's going to essentially live its life to eventually give rise to a person. No, it doesn't. I mean, everything that's needed for a cell to, develop into whatever it's going to develop based upon, the species it was present in, is entirely determined, number one, by its genomic content, but also based upon, more importantly, factors that were already present in the cell before it was fertilized. and to me, I mean, that's kind of surprising in a way that, it should be impressive to people that, that the kind of, very complex behavior of, cells to differentiate and to kind of divide labor among themselves by, differential gene expression and all sorts of various things, right? That it does it almost kind of spontaneously, that it has this kind of innate capacity to do that, and that it doesn't require anything that needs to direct it, really.
So, Stupid Horror Energy said, is ATP synthetase a machine? That one is really tempting to call it a machine until you start thinking that it is self-organizing. So it's more like a dust devil or a tornado than a machine.
I mean,
Isn't it ATP synthase?
Synthetase, yeah, that little motor, the little, the thing that spins around, and it's got a rotor and it kicks out ATP. On the
synthase.
I think it's synthetase.
I think it's synthase. I thought it was
synthase.
Trace.
Yeah, it is ATP synthase.
Anyways.
Look up synthetase to see if there were two different, but yeah, it's eight. But it was probably just a typo.
Yeah.
Is it?
I'm being pedantic.
Oh, it's called a synthetase. Yeah, it is called synthetase.
Oh, maybe it's a different ace.
They also synthase. Okay, there's a synthase. That's kind of a different definition, I think.
I thought there was only synthase.
Yeah, I
there's-
looked up ATP synthetase and I am finding absolutely nothing.
Are you kidding?
Nothing.
Oh,
It said showing results for ATP synthase. Let me see. Search instead.
There is, like, on the Merriam-Webster, synthetase.
Yeah, and I've always I've seen okay, right here, amino acyl tRNA synthetase.
Now I'm finding it.
Oh, that's what I'm confusing it with.
Synthetases are different classes of enzymes.
Yeah, what is the difference?
Synthetases are responsible for the synthesis of a molecule. Typically.
Okay, now.
Oh, it is. You're right. You pedantic. I'm putting you in the back room, Andrew.
Well, I gotta head out soon anyway, so.
You got to pay to get out of here.
Nice knowing you guys.
Oh, SWE said it's called. I swear I've seen synthetase. Maybe people, do
Maybe
it either way.
Yeah, I've seen synthetase too.
Yeah, I think so.
That's interesting. I've never. I haven't seen that been used too much, but okay.
Synthase has never crossed my mind. It's not in my 70-year-old store of memories. But there may be a reason for that, because they're kind of falling out. So, every day I scrape memories off my pillow every morning. I just dust them off. I don't need that one. Don't need that one. Don't need that one. I no longer record Netflix or Amazon Prime. I only record it as long as I'm binge watching the series, and then I discard it all. That way I can watch it again 2 weeks later and enjoy it just as much. Pretty cool. Yeah. Saves on streaming costs. Anyway.
I had a small update.
Mm-hmm.
How's my audio coming in?
Pretty good.
You're clear.
All right, cool. Well, I had a small update about our conversation with Standing and Company the other day on genetic entropy. Because I was thinking about our conversation, and so you had Redefined Living asking us, how do you account for all these new genetic diseases that are rising in the population if it's not genetic entropy?
Yeah, did you check that out?
Well, I replied back to that by saying that if you search through the literature, the reason that they're giving for this is relaxed natural selection, that selection has been muted in the human population through industrialization and healthcare. And Standing responded to that. And it was his response was interesting because he basically agreed that relaxed natural selection is a thing. And he gave this example of a zebra in the wild with a short leg versus a human with a short leg, and how the zebra is zebra is less likely to survive. And then he says that, but this is trumped in humankind by intragenic competition, by all that warfare and all the killing that we involve in and stuff. So that was his account. But the interesting thing about all this is that I was saying relaxed natural selection is how we should interpret, the rise of genetic diseases and stuff like that in the population over time. His account on that is no, it's not simply relaxed natural selection, it's genetic entropy. But then he turns around. What's that? Go ahead.
He was claiming that wars would counter that, right?
Exactly. And so when you're saying that wars counter that, what you're saying is that intragenic competition trumps genetic entropy. Because, I'm saying it's relaxed natural selection. He's saying it's genetic entropy. But then he turns around and says it's this thing that we're disagreeing how to interpret, whether it's GE or relaxed natural selection, whatever it is, it's being trumped by intragenic competition. And so he's actually giving an account that goes against genetic entropy, saying that intragenic trumps it.
Yeah, he doesn't care.
So, but the only thing, though, is that it's only a minor update because I responded to that, too, because I disagree with it based on the stuff that I gave about Steven Pinker, that the evidence right now seems to be, and I checked this out, and there are several articles supporting this, that overall, globally, human violence is declining. And so if that's the case, then intragenic competition would not trump either relaxed natural selection or genetic entropy, insofar as we're talking about the accumulation of genetic diseases and stuff like that over time.
Yeah, there's something else that needs to be said about that. Now, one of the reasons why we're identifying so many thousands of new genetic diseases every year is because we have the genome and we're able to check them now.
We can identify them better.
Okay, there's two more points I want to make. The other one is cancer rates are actually falling. The other one is we're actually living longer, so these things are showing up. And another one is if you look at autism, we have widened our basically diagnosis of autism or being quote unquote on the spectrum. And it's turning out that is rising more rapidly in middle class white people than anywhere else. And it's probably because those are the people that are getting diagnosed.
Yeah. But the other thing
it's bullshit that we have more disease.
I'll leave once I'm done making this point because I got to go to bed soon.
Okay.
But
Me too.
The reason that probably I'd wager to say that the reason we're getting more updates on new genetic diseases and things like that are happening is, number one, the major reason is because of the presence of and the development continually of what's called next-gen sequencing that allows genomes to be sequenced like for fractions of a penny. That makes it so that we can have high throughput, and also very good quality on the types of genomes that are being sequenced. That allows you to make measurements on types of variations within disease and kind of abnormalities that occur within genetics and being expressed that way. So, I would say that's probably a larger portion of the reason that we're able to detect more genetic disease now than.
Yeah, and the other thing, we're looking more carefully and we're finding a whole lot of variation. And some of these, we're just looking a lot harder and we're recording a lot more. I don't think if the idea that we're getting more unhealthy, go to a mall or walk around the world and look at human beings and tell me that they're falling apart. They just don't appear to be. Huh?
The other reason that we're able to identify diseases as well is because it's been recognized that epigenetics plays as much a role in the kind of in how diseases work as much as it is the type of gene that you have. The absence or the damaging of certain methylase proteins, for instance, can cause overexpression of certain genes, and the hyperactivity of it can cause repression.
One way you could look at this is if you took everybody under the age of 24 or even under the age of 22 and looked at how many genetic diseases are in that crowd that are actually showing themselves. It's more than just having the gene. The gene has to express at some point. And I don't think that the human race is all that. The human race is doing fine if you look at 22 and younger. And that is what makes a difference for evolution. I mean, if they're trying to make a case that humans are degrading a bit, yes, that's what Kondrashov was worried about. But he couldn't make a conclusion either way. If you read the entire book, he doesn't make a conclusion. Leo.
Oh, I just wanted to say something that I pointed out to Sam, Redefine Living, like 3 times, is that genetic entropy, because I defined what genetic entropy was according to John Sanford's book, the guy who laid out the whole idea of it, and they agreed with it, that it's the reduction in fitness and eventual extinction of a population due to the accumulation of deleterious mutations. And they were like, oh yeah, 100%, that's what it is. And I said, okay, great. Well, none of the research shows that's happening. In fact, we've taken mutations, we've saturated viruses, and I even think some types of bacteria with a whole slew of mutations. And in some instances, I would argue in a lot of them, it actually increased their fitness. It didn't decrease it. So the point that I was trying to make was that once I pointed this out, they said, well, yeah, well, how do you explain all the accumulation of these genetic diseases to humans? And I said, well, it doesn't matter. And Sam was like, well, that's genetic entropy. And I, like 3 times, had to say, no, the accumulation of mutations is not genetic entropy. The decline in fitness as a result of the accumulation of the mutations is genetic entropy.
Yeah, he continually conflated that mutation accumulation versus genetic entropy.
Mm-hmm.
The last thing I wanted to say is there was also a point brought up by SFT and company that humans are more violent intraspecies wise than other species, and I took a look at that, and no, there's several other species that are more violent toward each other than we are. One of them is hyenas. There are several other examples that I can't remember off the top of my head, but you could easily like Google that and find the same articles that I did.
And also, can you imagine if like troops of chimpanzees had like nuclear bombs?
Yeah, exactly. That's it's not that we're more violent, it's that we have technology that when somebody decides to become violent just randomly, they can kill significantly more people than just like targeting one or a couple of people and fighting them. I mean, you can take a gun and go mow down a whole movie theater full of people. So it looks like technology where all these other species don't.
Yeah, you're breaking up again.
Of course I was.
But yes,
you just started.
I agree.
You just started. What if maggots developed nuclear bombs?
What if who?
Maggots.
Is there intraspecies competition, like violence in maggots? Is that a thing?
Well, they're pretty violent if they get on your body and you have some bad skin.
Or if they get like in your rice. That's not a fun one.
Oh, that's terrible when they get in your rice. But yeah.
Yeah, well, when you're stranded in the middle of nowhere and you need sustenance, maggots, grubworms, crickets, spiders. Oh yeah.
Well, I think my raccoons are evolving the ability to use cutting torches. I've seen evidence. Yeah, they're working on it.
Picture like a raccoon just.
Well, you notice they've already got the goggles on. They've developed the goggles, so.
It's kind of. I dig on raccoons. I had something that's kind of like it ties in to this whole thing about declining violence, but it's kind of irrelevant. So I just want to say it quick. It'll be my last thought, and then I'm gonna head back to the chat. But there's this archaeologist, Ian Morris, and he's written several books, like basically detailing the history of humankind since the last ice age. And he comes up with basically this Lord of the Flies hypothesis, where through the creation of states, this perpetual warfare actually leads to, because it is decreasing the level of anarchy, ultimately decreases the amount of violence. So the rise of nation states plays a role in the overall decrease in rates of violence for humankind, which would just be one account. I'm sure there's like a bunch of different reasons why. As a matter of fact, Pinker's analysis gives another reason why, in terms of like the advent of modern culture. But yeah, so like there are reasons to think that account that globally violence is declining is plausible, and I think that that's true, even though you might not think it. Like Leo was talking about, you take a machine gun, you go into the theater, you rip apart 25 people, and that's going to be blasted across the evening news all over the place, and people are going to get the impression about about how violent it is, and in certain areas. So
yeah, crime has been going down since what, 1989 or something like that, or 80.
Yeah.
Drastically. I mean, it went up in like the 50s and the 60s for some reason.
The thing is, even over a broader period of time, with the advent of the Industrial Revolution and everything, violence started to decrease as standard of living started to go up.
Yep, I think all that is plausible. So yeah, thanks for letting me give my little tidbit there, and I'm gonna head back to chat and keep listening.
Oh, thank you. Welcome anytime.
Thank you.
All right. Well, I'm gonna make out like an electron and tunnel out of this room. So you guys have a good night.
Nice.
Okay. I'm gonna make like an electron and tunnel out of this room, so you get and just cut out because you don't know when it's gonna happen.
Yeah, I think we should.
Like the Sopranos.
I think we should wrap up. It is Sunday. Some people have to work. I don't, but some people do.
I do have first day of class tomorrow.
I'm
Or first day back.
I'm doing my job right now. Holy shit, you guys!
Yeah,
Wow.
I'm really interested in your lab, your phylogeny lab.
Yeah, I'll let you know how it goes. I'm in the research lab, but I haven't actually started any research yet. So
yeah,
I'll be getting a research project hopefully together this semester, and then maybe working on it going forward.
Because I must be able to do some of this, and I must be able to use these tools because I have some things that I want to do. So
you're still pursuing a bachelor's, correct?
Yeah, I'm a second year undergrad.
Are you planning on going for a graduate level degree at all, or don't know quite yet?
I mean, I probably will. My bachelor's is in evolution and ecology, so I don't know what else I would do. I'll probably go for a PhD or at least some sort of grad program.
So Stupid Horror Energy gave me something to look up here. HIV and development of pores. Let's see, what is it? Where's the gated pore? Evolved right before our eyes. HIV one. Wow, cool.
Why won't she tell us what her credential? Like, she's got to have college, something.
Something.
She won't fucking tell us.
She's so sneaky.
Just absolute genius. Oh, cool. I found some stuff. Awesome. Okay, I will check into this. Molecular machines evolve easily, self-organizing. We shouldn't call them machines. We should call them dust devils from now on, or whirlpools, because they're very much like that. They're more like that than a machine. They don't really, like, whirlpools don't really make stuff for you. But
she said she's a bartender. She's such a liar.
She's a what?
She said she's a bartender. She's lying. She's got to be lying.
Yeah, pretty smart bartenders out there.
She could be a bartender with like a master's degree in like evolutionary biology or something. So
I mean, bartenders do make money. Like bartenders can make bank, dude.
Yeah, if you're good and you're in the right location. Oh yeah.
Most philosophers end up becoming bartenders.
I thought about maybe bartending part time just because I can juggle, so I could do like the cool bar flare tricks and stuff. I feel like that can make tips.
Oh yeah. You'd make bank. Yep. Okay, guys, let's end this.
Right.
Should we go home? We'll drive home. We're going to end this and drive home. I didn't hear agreement or.
God, I'm not driving home again.
We are going to drive this home.
We're going to drive this home
with one headlight.
With one headlight, right? Mr. Dylan's son had something to do with that one. I do like that song. There you go, 90s alternative, Leo. So you're a fan.
I believe it's Wallflowers that sings that song.
Wallflowers, yeah. What the hell's his name? What is his name? Something Dylan. Too bad I don't remember his name.
Bob Dylan?
No, his son. Jake Dylan, right? Isn't that his name? Or is it? Yeah, it's Jake Dylan, I believe. Anyway,
let's find
that is Bob Dylan's son.
Really?
Yeah, Jacob Dylan. Yep.
Yeah. Okay.
Jake Dylan home.
Does anyone want to help me end this thing? Does anyone have anything clever to say or some way to end this other than goodbye, cruel world?
You need to come up with a catchphrase to say at the end of all your discussions, Speed.
Or you know, I need to come up with some music. Like I don't know. There's got to be some damn thing.
Well, God, when are you going to start using OBS?
I'm working on it. I got so many things to do.
Just get a hold of Dave and have him teach you how to use his StreamYard hack and then get you started on OBS.
Peter offered to help me.
What's that?
Peter offered to help me.
Yeah. I mean, he can too. If you're using StreamYard, he'll be able to help you. But the thing that I've found out is that once you've got the basics of OBS down, learning it actually isn't that hard. Once you kind of know how to navigate the site or navigate the program and what certain things mean, figuring things out gets quite a bit easier.
Oh no, I can't bring it up.
Bring what up?
I'll get a copyright strike if I do anyway. Okay, guys, we're ending. Goodbye, cruel world. We'll see you later. See you tomorrow at 2 p.m.
Central Standard Time.
Yep. Goodbye.